Okay, so we now switch gears again. I hope your brains are up to this rapid transition between different fields, from switching the history of science now to back to genes and genetics and so on. And it's a real privilege to welcome back Professor John Bryant, who's spoken at a number of our Faraday courses. I wonder if someone could just close the door so we can keep the noise level down. Thank you. John has spoken a number of times for us at the Faraday Institute courses, and we're delighted to welcome him back here. John is an emeritus professor of cell biology from Exeter University, and he's been very involved in not only the whole field of cell biology and DNA regulation and so on, but also in the area of bioethics and also in writing books as well. And I hope you'll take a look at his book. I'm assuming it's on the bookstall, Our Life in His Hands, which the afternoon session will be slightly more relevant to direct links to that particular book. John's given a huge number of different lectures in different parts of the world, but I'm not going to take more time. You've got his bio in your handout. I'll simply hand over to John. He's going to speak on genes, makeup, manipulation, and modification. So thank you, John.
Yeah, thank you, Dennis, for a kind welcome. In fact, I think I've spoken Faraday Institute so often that I've got a season ticket between Exeter and Cambridge, or maybe not quite, but thank you for asking me again. Now, these next two talks, which unfortunately for you and for me, I'm giving both of them, they really begin to focus on what scientists do in the area of the new biology and how we use the scientific information in wider society. And I think very much that scientists involved directly at the lab, or also in the kind of ethical and social discussions that arise from that, Christians involved, really they do need the wisdom of the Holy Spirit in dealing with this material, because some of it is in fact raising questions which are rather fundamental to our understanding of what it is to be a person, whether it be genetics or whether it be stem cells. And so I hope you'll remember biological scientists in your prayers. Particularly those of you who have in your past truth practicing science. So we're going to have a quick look at these three topics. It will be quite shallow because there's a lot to cover here, and we can really only scrape the surface. So I'm going to first just ask us what is our genetic makeup? What do our genes do for us? I'm then going to think about how we might manipulate the genetic makeup of an individual by appropriate selection and deselection of genes. I then want to think about how we might actually physically modify by genetic manipulation techniques, moving genes, the genetic makeup of an individual human, and end up with a quick thought about eugenics, which Dennis and Bob both raised yesterday, and was anyway in this talk, so I thought it was a good time to explore that a little further and to see how far we might equate some elements of the new genetics with older eugenic practices. So, our genetic makeup. I'm going to show you a few faces that you will recognise who have got particular skills in particular areas of life. Just very quickly. One of those you'll recognise even more. And the question comes is how far do those skills and expertises for which we acknowledge these people, how far is that built in to their genetic makeup? And we can certainly say that without their genetic makeup, their particular abilities would not be present. And so, for example, as a runner, I will focus a little bit on Paula Radcliffe, where we can identify in her traits which are certainly in her genes. Absolutely, with no doubt about it. And that includes everything from her physique through to the way her muscles work and also the way that her metabolism works. We can almost certainly identify in Nigel Kennedy, we can identify part of his genetic makeup that is very appropriate for manual dexterity and for that sort of coordination. It's a bit harder with an author, but certainly there is a genetic element in our intellectual capacity. And I guess the question is, how far do we push that in terms of does that do for you, does that turn you into a cricketer, a musician, a writer, a runner? Now, how far far as genetic makeup go in, these are the cheerleaders for the football team at a university where our students have an exchange program. They've adopted Chanticleer, the cockerel in Chaucer's Canterbury Tales, as their mascot. I think Chanticleer's had a bit of genetic modification, though. I don't think he was ever that fierce. But about these cheerleaders, just have a look at those two there. They look quite different, and yet almost certainly the difference in them genetically between these two is no more than the difference between these two. So the dark-haired, dark-skinned girl and the two blonde, blue-eyed, typical American girls almost certainly have an awful lot of genes in common. In fact, Francis Collins suggests that we differ by 0.1% from any other human being, leaving aside the XY difference. I think actually it's slightly more than that. It probably goes up to about 1%. But on average, if I was to look at any other male in here of whatever race, the difference between us genetically would be between 0.1 and 1%. It's tiny. So those genes, in fact, specify what it is, therefore, to be a human being physically. But one of the pictures I want to make to you this morning is actually, we have to go a lot further than that when it comes to the makeup of an individual human person. As I said, we can identify in Paula Radcliffe all those physical traits that make her a runner, but they don't actually enable her to be a runner. They don't make her a runner. There's a huge amount else that went up into Paula's success, most recently in the New York Marathon, including her school, identifying her at an early age, Bedford Athletic Club, seeing the young talent and encouraging it, the support of her parents, the determination that she has in her personality. And so those things do not get specified by genes. The environment had a huge amount to play in her success. And more to the point, there are actual elements of our physical being that lie beyond our genes. So, for example, if we were to look at the way that our neurons connect, and the number of possible neurological connections in the brain is greater than the number of atoms in the universe, and that is 10 to the 80. 10 to the 80. If you think about the number of neurons we have, which is 10 to the 11, and the number of possible connections between each individual pair of neurons, you get a very large number. That cannot be specified by our genes. We know that that is something that changes over our lifetime in learning experiences and with the experiences of life. More dramatically, my wife works with marginalised and underprivileged people in Exeter, and showed me some research which has been published in Nature and elsewhere, showing that children who are desperately emotionally deprived in their early years, their brains fail to develop fully. And some of the most classic cases have been those orphans in Romania who have been left without any human contact. And so there we have, if you like, an emotional situation that is affecting the physicality of the brains and all that goes with the makeup of the brain. So although genes specify what we are in terms of much of our physical development, even when it comes to the structure of the brain, other factors will modify that. So I will go very strongly against the idea that we are completely determined by our genes. So in my view, it is not all in our genes. There's much more to it than that. But on the other hand, we need to think about what do genes do, what do genes do for us. And a number of people have got different views on this. This was the first edition of Richard Dawkins' The Selfish Gene. And his idea is actually it's not what the genes do for us, but it's what we do for the genes. The idea of the selfish gene is that we are just here to replicate our DNA, and the book is, and it follows on from then that genes determine what we do in order to make sure that the genes get replicated. And the book's gone through a number of iterations. I find it interesting that this series of images on the front includes a chimpanzee and also the Roman viaduct at Pont du Gard down in Provence, plus a little egret. And all these things are clearly built into our genes. The Romans, it was their genetic makeup to build a viaduct at Pont du Gard. And then, sorry, this is a rather fuzzy image, lest I forget. And this is the 30th anniversary edition, a very widely read book. And I think it's indicative of the influence that's had. I'm giving a talk in Brighton next week under the title, Are Dawkins' Genes Selfish? Now, there are thinkers, and this is the philosopher scientist John Dupre, who is also an atheist, incidentally, who will criticise Dawkins very, very strongly on the grounds that there are so many parts of our development, emotional and social development, that cannot be ascribed to our genes. And he talks about the origins of morality, conscience, and that sort of thing. But of course, the Dawkins School have got to reply to John Dupre. Well, actually, there are other things that we've got to take into account, and those are called means. And that's Richard's idea that in addition to there being the physical inheritance of genes, somehow there's an intellectual inheritance of social norms called means. Now, I always feel about Richard here that for somebody who is so keen on only accepting things for which there's evidence, that to propound a theory for which there's no evidence whatsoever, it seems to me to be a very strange contradiction in terms. But it's Richard's way of dealing with the John Dupre criticism, that there are clearly many elements of our being that are not directly heritable, and so there must be some other way of passing on this kind of core knowledge. Okay, so some partial answers to what our genes do. A gene is effectively a coded recipe, and if you like, what happens when a gene is read is that the cell will read the gene and it will go to its various store cupboards inside the cell, pick up the ingredients for the recipe and build them, and it builds things called proteins. The technicalities don't need to worry us, but a gene is a recipe for building something in a cell. One gene, one recipe. And so a genome, that is our collection of genes, is like a little library. And in the human genome, there are 23 books. And we've got 2 copies of each book. Each book is a chromosome. And men and women actually differ because women have got 2 copies of a book called X, and men have got 1 copy of a book called X and 1 copy of a book called Y. But if we think our genome is therefore a set of recipes, and I've suggested that we're not just the product of our genes, and therefore I will be very, very strong in saying, please beware of the genes are us, rather than Toys R Us, the genes are us fallacy. Because although genes help to make us, we're not just a bag of genes. Now, I'm going to be a bit technical, and this is where the biologists can chill for a few minutes, because I'm just going to give a bit of a technical background here. So if we were to look at a gene, we'd find it was made of this wonderful molecule, my favourite molecule of all time, called DNA. I've been working with it since I was a postgraduate here in Cambridge, about 100 years ago. Actually, that was before Watson and Crick, wasn't it? And the genes are built of this material called DNA. And if we were to look at these long molecules in the cell, we would be able to find a specific tract, a specific instruction, a specific recipe, which we're going to call a gene. And the genes are arranged in long strings called chromosomes, and each of those chromosomes I've called already a recipe book. And the collection of chromosomes is our genome. And the code is written into the order of the building blocks. So the building blocks are quite simple. There are just 4 of them. But because the molecules are so long, the code you can write into them is immense, it's infinite. And it's read in 3s, and biologists will recognise these 3 here as being appropriate for starting off a protein recipe. And this is just a tiny, tiny, tiny section of a gene. Most genes are the order of thousands of these building blocks of an individual recipe. But here's a thing that is not widely thought of, but it's the code is universal. What does that mean? It means that every single living organism on the planet reads the code the same way. So 1 or 2 organisms are able to do a little bit of editing of the code, but in general terms, every organism on the planet reads the code the same way. And I like to think in evolutionary terms that every living organism from which modern life has descended has read the code the same way. If we didn't read the code the same way, genetic engineering would not be possible. It would not be possible to take a gene, a recipe, from one cell and to put it into another. And I'm going to think a little bit about genetic engineering later. A universal genetic code that unifies the whole of life. And biologists have no trouble with Bishop James Jones's statement yesterday that we are part of the living environment, because we recognise that we have this affinity at the genetic level with everything else that lives on Earth. And this is more or less how genes work. This is just a quick cartoon what you imagine, this is a long string of DNA, and it's built always of those building blocks, but particular sections in that long strings provide us with the recipe, the coding sequence. We always keep the gene back and make a working copy of it. It's a bit like you might have a master copy of plans, but in order for the builders and the architects to take them out onto the site, you make photocopies. This is actually what happens in the cell, that always a working copy, a messenger molecule is made, which copies the coding sequence. And that provides the recipe, which is translated into one of these structures called proteins. And the proteins have got specific functions in the cell. So that's what genes are doing. They're providing recipes for building molecules called proteins. And those proteins affect what our cells do, and building the cells together affect what the tissues do, affect what organs do, affect what the body does. Now, although we want to be a little bit, we want to be very fuzzy in terms of the role of genes in the makeup of our personhood because of the influence of all the other factors, what we do know is that there are some situations in which we can identify what happens when that sequence of building blocks changes. We call those mutations. It simply means change. And as somebody said yesterday, since it's almost certain that we started as dark-skinned people in East Africa, we white-skinned people, we Caucasians, are in that sense mutations. But actually, we're all mutations. We're all mutants. Because we all vary genetically from each other, there must be, in some senses, in which we're all mutants. But some of these mutations have directly identifiable effects on us, on what we call the phenotype. The phenotype is the way that the genes come out in us. And some of those changes are obvious, like, that people are unable to make melanin in their hair or blondes. I used to be able to make melanin in my hair, but that's an age-related change. But in some cases, the condition that ensues from the change in the gene, we actually regard as a disease. The person is, if you like, less fit for life. And so we have heritable diseases. And they've been known a long time. Even the communities that just sat down and thought about things, like the Greeks, recognized that there were traits that went in families. They didn't understand why they did. But occasionally you had mutants of the dark-haired Mediterranean people who had red hair. And that was a very dominant mutation. It traveled in families. In a lovely story told by Professor Steve Jones, a rabbi in AD 200, circumcising a boy, and he bled to death after circumcision. And then his brother later bled to death after circumcision. And he surmised that there was a condition in the family, and so all the boys in the family and the cousins were then excused from, or subsequent boys were excused from being circumcised. Because what he didn't know was he'd identified a disease called haemophilia, extinct haemophilia. So we've known about heritable diseases for some time, and a lot of the emphasis on human genetics from the beginnings of the 20th century have been on understanding the inheritance of these conditions which travel in families. And obviously, if the mutation was caused in a single gene, then that was so much easier to understand and follow. And there are about 4,500 of these single gene disorders. I'll talk a little bit later about some of the more complicated conditions. Indeed, well, maybe not. I'll talk about it now since I've brought it up. But Francis Collins, the director of the Human Genome Project and a practicing Christian, believes that nearly every illness we can imagine has got some genetic component. That it is true that some people get colds more often than others. It is true that some people are more susceptible to viruses because there's a genetic component to our immune system. So there's an increasing feeling of susceptibility. And some of that susceptibility comes from interaction of our genes with the environment. And so some environments will trigger increasing susceptibilities, maybe working with pollutants. So it's quite complex. But single gene diseases are the ones which get the most attention. So remember that what we're talking about here is always, with single gene diseases, a change in something, a change in the DNA. It might be a change in one of the building blocks, it might be a deletion, it might be an addition, but it's a change in the way the building blocks are arranged in the code. And so we have two of the well-known recessive diseases where you need in the damaged copy of the gene to be in both sets of your chromosomes. You need it to be both in the maternal and in the paternal. So both sets of chromosomes, and only if the mutation is present in both sets will you get the condition. Cystic fibrosis is very common in North European Caucasians, and particularly in Celtic-derived lineages. In this part of England, the carrier frequency, the frequency with which exists one mutated copy of the gene, is 1 in 25, so that statistically there's at least one carrier in this room, statistically, but not showing the disease because you need two copies. Down in Cornwall and in Brittany, in Northern Ireland, the carrier frequency goes up to 1 in 22. Now, work out the likelihood of having a baby at 1 in 25 with somebody else with that mutation. Then each of your fetuses has got a 1 in 4 chance of having cystic fibrosis. So it is the commonest condition in North Europe that is a genetic condition that's regarded as an illness. There is a commoner one, which is a metabolic dissimilarity. Sickle cell anemia, we've talked about Celts here. Sickle cell anemia is particularly associated with people of Afro-Caribbean descent. And so in the black population of the States and in West Africa, Nigeria would be an example, this is a common illness. Thalassemia is common in the people of the Eastern Mediterranean. Thalassa, from the word for sea. And then we have dominant conditions, and in dominant conditions you just need one mutated copy of the gene. So it doesn't matter which side it comes from. If you've got one mutated copy, you get the illness. Huntington's is a particularly unpleasant neurodegeneration. The onset's in your 40s. So you pass it on to your children by the time you know you've got it, unless you had a genetic test. And then there are X-linked diseases where the faulty gene is on the X chromosome, and women, you're absolutely fine because the correct copy on the other X chromosome will override the faulty copy. These are recessive, effectively. But us guys, we've got problems because our pathetic little Y chromosome with its handful of genes has not got the gene that's mutated. And haemophilia, Queen Victoria's gene, is the classic example of this, and one discovered by the rabbi those years ago. So part of the motivation for the Human Genome Project, was set up to determine the building block order on each of our 23 chromosomes in an average human being. It's actually a mixture of many human beings. I think Craig Venter has had his genome totally sequenced as an individual person. The idea is that if we determine the sequence of the building blocks in all 23 chromosomes, we get some sort of insight into these genetic changes that occur to cause disease, into the genetic changes that occur to cause predisposition to disease, and also into the genetic changes that are not heritable, but occur within the lifetime of an individual to cause things like cancer. So some cancers are heritable, but most are caused by genetic malfunction only in the lifetime of the individual. The Human Genome Project was set up to have a look at these things, to get a handle on genetic disease. It proceeded extremely rapidly. The first draft occurred just 10 years after the project started. I need to say that quite a lot of human genetics was going on before the Human Genome Project. If you read American accounts, you kind of think that America started human genetics, but I had appointed a young colleague back in 1987 who was working on sequences of some genes on chromosome 19, and that was clearly before the project started. She was later headhunted by the University of Newcastle for a professorship. So there was human genetics going on dotted about the country, dotted about the developed countries of the world. And really the Human Genome Project started to draw these things together and very much increased the funding for this sort of work, a huge increase in funding. And we've got a pretty good picture by 2003. An ethical thing that we haven't got time to talk about now was there was tension between the public and the private sectors because Craig Venter's company wanted to patent the sequence of every gene that they found, and publicly funded labs said, no, this is public knowledge and should go to public databases. And so the Sanger Centre down the road here and Hugo in Paris were putting the genes in the public databases as soon as they were kind of coming off the sequencing machine. to make sure that the vector was stylish in doing that. Some genes have been impacted, but on the whole, this is publicly available knowledge. And I think for me as a biologist, this still remains the most amazing fact of all. It's come out of the Human Genome Project. If you look at the average mammal, how many genes do you think it'll take to run a medium-sized mammal like a human, or a small mammal like a mouse, or a big mammal like an elephant? The numbers come out about the same for all of us. They come out at about 80 to 100,000 when you look at the individual functions that our cells perform. And yet, try as we might, we've only come up with 25,600. And it must mean that genes are cleverer than we think. That there must be, that we know now there are genes who've got more than one job. We call them moonlighting. They have another job. And in my lab, we accidentally discovered a moonlighting gene some 12 years ago, which provided nearly all the grant funding for the last few years of my active research life. And I think, for me, the idea that we now need to work out how this small number of genes runs a small mammal on the physical side is really quite a challenge. It's wonderful. And so it was the end of the beginning. But nevertheless, it's given us a real handle on a number of these genetic conditions. It's given us the ability to test for a very large number of diseases where we can now put down the disease to a genetic change. We can search for that genetic change in the DNA by molecular methods, which can take place very quickly. You do not anymore have to wait for symptoms. Has my child inherited cystic fibrosis? I don't know. You've got to wait to see what happens when the child's born. But now you can test the DNA. You don't need to wait for symptoms. Will I get Huntington's when I'm 45? I don't know. Actually, you can find out now. And this means that we've now got a way, if you like, of selecting for and against particular genetic conditions. We can manipulate our genetic makeup because we can test individuals at almost any stage of life. And we'll think a little bit more about what that means practically in a family in a moment. We can test adults. We can diagnose conditions which have suddenly appeared. So, for example, it might be that in your 20s you start getting muscular weakness, and it turns out that the genetic diagnosis shows you've got a form of muscular dystrophy. You might be concerned that you will have Huntington's later, and you have this kind of feeling, well, I'm just waiting for the disease to happen. I'm a pre-patient, and what that does to your life. You might be interested to know whether you're a carrier. Have I got a single copy of the mutated gene? And does that affect my family and marriage planning? There's a particularly nasty neurodegeneration called Tay-Sachs disease, which is very, very common in people of Ashkenazi Jewish derivation. And it's devastating. A child will begin to just disintegrate, basically neurologically, from the age of about 7 onwards, and will usually die unpleasantly by the time they're about 12. And in the Jewish communities in Chicago and in New York, the rabbis started, so once genetic tests became available, started to keep a record of individuals' genetic status with regard to carrying Tay-Sachs. And he just kept the record. And when young people fell in love or planned to marry, they would go to the rabbi to find out whether they were both carriers. So these were tough decisions being made. And he would advise them if they were both carriers. This is the situation. That if you have children, each child's got a 1 in 4 chance of having Tay-Sachs. Some didn't marry on that basis, which is a tough decision. Others decided not to have children. And the incidence of Tay-Sachs in the Jewish communities of New York and Chicago, and now in Israel, where the Ashkenazi Jews have started to migrate back, has decreased quite remarkably because of this. The program is called Eugenics with the Smiling Face. And we can diagnose children. If your child starts to go down with something that might be an inherited disease, you can now test them, and that may well affect the family dynamics. If you suddenly find you've got one child with cystic fibrosis and the other one hasn't, how does one treat the 2 children? We can test newborns. Now, of course, all newborns are tested with the heel prick test, and until a few years ago, that was only for one genetic condition. That was phenylketonuria, and you didn't do it by genes, you did it by biochemistry. Now, in most local authorities, 5 conditions are tested for, and it's DNA rather than biochemistry. And those conditions include phenylketonuria, they include cystic fibrosis, they include an inherited form of hepatitis. In the Greek Cypriot communities in East London, they include thalassemia. But 4 or 5 conditions are now routinely tested for genetically. But in the film Gattaca, all newborns were tested genetically, and it's a wonderful piece of science fiction with a nice sociological commentary, where the hero of the film had not been born by IVF as most children were, but had been born because of a fling by his student parents in the back of a car, and was born by the normal fun method, if you like. But his barcode showed that he wasn't perfect. He was called an invalid. Very nice pun. And the whole film was about him trying to fool the authorities about his genotype to get onto the spaceflight program. And Gattaca means nothing. It's simply the 4 letters of DNA turned into a word. But a great film to go and watch. Raises some very interesting issues, and I know quite a lot of sixth form school teachers who show it to their students, including my oldest son. Okay, you can test newborns, you can test fetuses. The baby's got genes clearly, and so you can test fetuses. And I'm told by physicians that we are getting more genetic requests for terminations, and some of those are for very trivial conditions which might otherwise be managed. Phenylketonuria is the classic example. That you can manage this by diet and lifestyle. The diet is very boring because you need to avoid a particular type of protein. But you could manage it, and yet people are now asking for terminations of phenylketonuria if it's known to run in the family. And we can test pre-implantation embryos. That is, embryos that have been generated by in vitro fertilization, but they have not yet been placed back into the uterus. We can test them to look at their genetic makeup. So here's 1 or 2 of the tests. This is the prenatal test, where guided by the ultrasound scanner, the nurse and the technician are inserting a long needle and pulling out some of the amniotic fluid which contains the baby's cells, and those cells can then be read for the particular gene that the parents are worried about. And then pre-implantation diagnosis. It depends very much on the remarkable developmental plasticity of early mammalian embryos. Here's a newly fertilized human egg, and I have to acknowledge, as I always do, the Biochemical Society. These were published in the Biochemist and came originally from one of the IVF clinics at St George's Hospital. This is a newly fertilized human egg, and the 2 sets of genetic material are here and here. They've not yet merged. The sperm has gone in, but it takes about 10 hours for the genetic material to merge, and in fact it doesn't merge properly until after the first division. And then we go through this series of cell divisions, taking about 7 days, as in nature the embryo comes down the fallopian tube. This of course is all happening in the petri dish in the picture, until you reach this stage here, the blastocyst, and we're going to hear much more about the blastocyst in the lecture after lunch. But at this stage here, these 8 cells, there is no up, down, left, right, no axes at all in the early human embryo. The cells here are not committed to any particular developmental pathway. Unlike the fruit fly, where even the egg has got a front and a back, a left and a right, and an up and down. The egg has got polarity. In humans, there's no polarity even at this 7-day stage. These cells do not know what they're going to be, and so much so that you can pull one of them out, and the 7 cells remaining behave as a normal embryo. So we can test early embryos for genetic mutation before, and then decide which is going to be placed back in the womb. And here is the test. This is quite blurry because what we're doing is we're looking down a microscope at an embryo being kept in its nutrient solution, and it's being sucked against a fine pipette, and a cell is being pulled out. These are quite big cells. The whole embryo is about 0.1 of a millimetre across. So it's pretty small, but the cells in normal are much bigger than normal cells. So you can pull one out and test them. And some people who are at risk of genetic conditions running in the family find that PGD is more acceptable to them than setting up, than having a pregnancy and then terminating it. The idea of selecting an embryo, selecting against an embryo, not implanting it, they find more. And this is actually what's happening, that if you're at high risk of a genetic illness. Couples are selecting PDD. And also you'll be aware that in some cases people have selected embryos so that they can donate cells to an older child who's ill, the saviour sibling scenario, and you might want to ask about that in question time. And all this in Britain is regulated by the Human Fertilisation and Embryology Authority, and most recently they've added to the list of things you can be tested for late onset conditions, conditions that don't affect you till later in life, and also high susceptibility conditions, conditions where you have a high chance but not an absolute chance of having the illness, such as some forms of breast cancer and colon cancer. Now I'm going to show you a video clip which illustrates better than I can pre-implantation diagnosis. The fact I'm showing this will extend the lecture very slightly, I'm sorry, Chairman, keeps people interested. So just look out for the issues that are raised by this, and let's see if we can get this to work. Wonderful. That often happens. Let's try a refresh here. I was hoping actually to clip this into the PowerPoint presentation, but the New York Times, where it comes from, blocked the download, so I'm not getting anything at the minute.
Might just be downloading.
It blocked me saving it. Come on. It was working at coffee time beautifully.
Shall I get Bryant?
No, I'll talk people through it if necessary.
But I'll leave it to download, John, and then keep talking and see if it downloads what we're talking. It might just be time for downloading.
No, it's normally much quicker than this. I'll give it one more go, and I'll talk you through the video otherwise. Let's forget it. What? Oh, no, something's happening. Something's happening.
Yeah.
There we go. That's a word from our sponsors.
Every day, it seems, talk of oil, energy, the environment. Where are the answers? Right now, we're the largest producer of clean, renewable geothermal energy in the world, generating enough energy to power 7 million homes. Imagine that. An oil company as part of the solution. This is the power of human energy.
So, enjoy my new side?
Yeah.
Okay.
Chloe Kingsbury is a perfectly healthy 2-year-old child with 2 adoring parents.
You want your shoes? Okay.
Chloe lives with her mother Colby and her father Chad in a modest suburban home outside of Chicago. Like most new parents, Colby and Chad wanted their child to be brought into this world with all the hope and promise that other children have. But there was one problem.
In my family, there is a cancer gene that has devastated our family. It's killed my mother, my 2 uncles. My grandmother's lost all 3 of her children. I have 2 cousins that have been affected already with different forms of cancer.
The gene Chad is talking about can lead to a form of colon cancer that can affect the person carrying it in midlife, usually around 45. He tested positive for the gene and learned that, according to the laws of genetics, there was a 50% chance the gene would be passed on to his children. Many people with the gene never get cancer at all, and if the cancer is caught and treated early, there is a nearly 90% survival rate. Still, that level of risk proved too high, given the alternatives available.
No matter how remote the possibility is, it's still a possibility that she could get cancer. Why burden her with that if we don't have to?
From a relative, the couple learned about a medical procedure that is becoming increasingly common, whereby aspiring parents undergo in vitro fertilization and select only those embryos that are lacking in certain potentially harmful genes. The procedure is known as PGD.
PGD stands for pre-implantation genetic diagnosis.
Dr. Kenneth Offit is Chief of Clinical Genetics at Memorial Sloan Kettering Cancer Center.
And what PGD does is to take an embryo at the 8-cell stage very early, to take one of those cells out, test it for genetic mutation. If the embryo does not have that mutation, that gets implanted and goes on to become a fetus.
But for many couples like the Kingsburys who decide to go ahead with PGD, the decision isn't easy.
My wife and I did a lot of reading, a lot of research. We talked about this for 2 months because it's a very difficult decision to make. But at the end of the day, I think having a healthy child kind of outweighed everything.
Part of the difficulty, says the couple, was bringing others on board to accept the idea of meddling with nature. That meant overcoming some religious concerns from Mr. Kingsbury's family and convincing friends that what they were going to do was not something out of science fiction.
Trying to explain this process to them that we ourselves didn't fully understand just sounded like, some type of space age movie, and they just thought, why are you doing this?
The procedure required that Mrs. Kingsbury undergo frequent hormone injections and a painful egg extraction process.
It all seemed very complicated at first. There was just a lot of shots, a lot of medicine, a lot of procedures involved.
And to pay for the procedure, the couple also had to dig more than $10,000 out of their savings, a fact that weighs heavily on the doctors who were part of this revolution.
One of the issues that has struck me from the outset here is that we need to be fair. We have to be equitable in the distribution of these technologies. If we decide that we're going to go ahead, for example, then everyone should have access. Right now, to do PGD, it can be $10,000 to $15,000, and the insurance coverage for this is very variable. They may cover it if there's an infertile couple, but generally will not for these kinds of elective decisions that we're talking about.
PGD has been around since the early 1990s, but as awareness of the procedure has spread, more people than ever before are making this choice. Dr. Offit suggests that part of PGD's allure has to do with a desire among couples to manage their own destinies, rather than let nature take its course.
Over the last several years, I have become increasingly sensitive to the fact that many of these young people do feel a sense of empowerment, of taking control, when they realize that they can, if they choose, have children who are guaranteed not to carry these mutations that they have carried, that their parents, their grandparents, and on back many generations.
Testing for our children was the one actual active thing we could do. Up until then, we were just watching. We were just watching cancer take his family over.
About 1 to 200 Americans carry a genetic mutation that makes them more susceptible to breast or colon cancer, the two common hereditary cancers for which there are now genetic tests. But as scientists learn more about the genetics of cancer and other inherited traits, the dilemmas become more complex.
The other ethical challenge and question that has been brought up is referred to as the slippery slope. If we're going to make it possible to select for certain genetic traits, who decides? But I do recognize that as we come to better understanding of other polygenic disorders, such as obesity and other traits, then it will become murkier. And I think that is a focus that will require a very concerted social discussion.
That discussion is already taking place. But in the meantime, couples like the Kingsburys are pressing forward with their desires to have children free of the diseases that have burdened their families. The Kingsburys, in fact, are planning on having a second child.
Now that Chloe's here, everybody adores her. And, we're going to do it again. We want to have another child, and we're going to do it again, and everybody is fully on board this time.
Okay, let's just get the PowerPoint back up and running. Quite thought-provoking, though, wasn't it? Right. You notice that it was a late onset condition. The penetrance was not 100%, that you didn't necessarily get the illness with the gene, and there's a 90% remission rate if it's detected early. Nevertheless, PGD was undertaken. Currently across the world, there is nearly 1,200 tests currently available for direct genetic testing, and there's another nearly 300 available at the research level. In the UK, of course, we have social medicine rather than medicines paid for by your insurance company, and the HFEA currently authorises about 60 genetic tests that can be undertaken on the NHS, and you can find a list of them, a list of most of them on the NHS website. There's a very small number that aren't there because they're so rare that it would identify the families if the condition was listed on the website. So the HFA will tell you how many PGD tests have been undertaken in the UK and conditions that will be tested. But as the doctor hinted, there are questions about what else should we be allowed to select for. Polygenic things, where a number of different genes interact with each other and maybe with environments to produce conditions. What about non-medical conditions? What about genes that affect our appearance? How far should we manipulate, if you like, the future generations by selecting genes that have got nothing to do with medicine? And so here are some of the issues that arise. That we're seeing in some elements, some areas of geneticization in medicine. The genes have spoken, and that's all you can do about it, hence terminations of pregnancy. We wonder whether we're narrowing down our concept of the normal, sort of a kind of genetic version of Freudianism. Does it reinforce the idea that we're just a bag of genes? And what about commercial attitudes? The American doctor spoke of insurance, and it's certainly clear that some American medical insurance companies are refusing cover to families where genetic conditions run in the family, unless the family opts to have termination or pre-implantation diagnosis to avoid the birth of children with particular genetic conditions. What about employment? And so a number of more general issues arise too. Who should know about these things if you've got a genetic condition? And in your papers, I've thrown out a series of case studies that enable you to think through some of the issues and become concerned and worried about them as well. Okay, so that's manipulating the genetic makeup of a future human being by selecting for and against particular genotypes. And it inevitably raises the issues about, are we rejecting people by rejecting embryos? But can we go further than that? Could we actually change the genetic makeup of a future human? Would it be possible to add or take away a gene from a future human being? And this is based on the recombinant DNA technique, where we can identify a gene, we can splice it into a circular DNA molecule, sometimes made in the lab, sometimes obtained in nature, so our molecule's got the gene we want, and then we can put it into an appropriate cell, and that essentially is the basis of all genetic modification. Taking the gene we want, putting it into a carrier DNA molecule, and getting that DNA molecule into the host cell, whether we're talking about bacteria or plants or animals. The basis of the technique is the same. I always like to show this. These are real DNA molecules in my lab, in the electron microscope. And you can see that they are circular, and these are actually recombinant molecules, but you can't tell that because you can't read the base composition on the electron microscope. But there they are. They don't lie perfectly flat in the EM, so they're never perfect circles. And so here is a mouse egg, a newly fertilized mouse egg, having genetic material added to it. And this is one way we could modify a future human being. We could take that newly fertilized egg and put the genes in. Genetic modification via the early embryo. For small mammals, there's another route as well. We can use stem cells, but I won't talk about the technicalities of that. This particular route would be appropriate for large mammals, such as farm animals, such as humans. We can add the DNA to the fertilized egg. And for reasons that I think still we find as scientists quite incredible, or maybe even puzzling, often cells will take up DNA that you inject into them. It's really very strange they should do that, but they do. So, should we be allowed to genetically modify humans? It's normally discussed in relation to gene therapy, and because genetics shows us that some genes go wrong and cause diseases, can we correct the faulty gene if the person is born with a problem? In other words, can we take a pre-existing human, a child, for example, who's got a genetic illness, and see if we can replace the faulty gene? Now, of course, we've got 70 million cells, and all except our red blood cells, carrying 2 copies of our genome. you know, that if the person's already here, how do you get the DNA into all those cells? The answer is you don't. You just look at the cells that are most affected by the condition. So in cystic fibrosis, we're thinking mainly of the epithelium of the lung. For immune deficiency diseases, we're thinking mainly of the bone marrow. And these are cells that can be targeted rather more readily than a much more general condition. And so here are some conditions which have been corrected by gene therapy. Cystic fibrosis, not very successfully. There has not been any long-term remission. Severe combined immunodeficiency disease, the boy in the bubble illness, where their immune system is so compromised they catch anything that's going. There have been 12 attempts at gene therapy, changing the genes of the bone marrow. 12 attempts in Britain and 11 in Paris. All the British, 11 out of the 12 in Britain were successful, completely successful. And in fact, the oldest child is now 6 and is at normal primary school, suffering all the buffeting that you get. In Paris, 3 of the patients suffered a side effect. A leukemia-like disease occurred after the gene therapy. And when Greg Ormond Street compare their technique with the Institut Pasteur, it's very hard to know why the Parisian system resulted in the side effect. The two differences in the technique look trivial, which shows us that there are things in science that we still need to puzzle about. The differences in the technique are tiny, and you cannot understand why one has induced side effects in 3 of the kids and the other one hasn't. And then more recently, there's been one successful cure for congenital blindness by gene therapy. And then in the States, we've now got these are curing non-heritable but nevertheless genetic malfunction conditions such as cancer. Non-heritable cancers, but which are caused by malfunctioning of a gene in the body. And there have been successful cases now in the States of switching off the rogue gene that is malfunctioning in the body by something called RNA inhibition. And again, technically we don't need to worry about it. So gene therapy is here and has had some success. Now, wouldn't it be nice, you think, to try to eliminate a genetic condition completely from the family line? That would be called germline therapy. And in other words, it would be heritable. We would change the makeup of a future human being so that change went down through the generations. And actually in Britain, our law forbids you from doing it. The law will allow you to experiment on genetic manipulation of embryos, allow you to do that. It will not allow you to put those embryos into the womb to establish a pregnancy. So even if we think germline gene therapy is a good thing, it's illegal because the embryo would have been genetically modified. So that's in contrast to genetic selection, which is, as we've just seen with the video, is legal both in the UK and in the USA. Interestingly, I reckon if you went out into the streets of Cambridge and talked to them about what germline therapy was, they will be supportive of the idea. That's a great idea, eliminating forever in a family line, particularly genetic disease. The scientists may well reply, actually, with pre-implantation genetic diagnosis, we probably don't need to undertake germline gene therapy. But of course, once we start thinking about gene therapy, we also might think about genetic enhancement. You know, what features would you like your kids to have that are going to make them more successful in life? Maybe in terms of their looks, maybe in terms of athletic prowess, maybe in terms of playing the violin like Nigel Kennedy. Can we identify genes that might make them more successful, more attractive, etc. in life? Heritable enhancement. And I think actually when it comes to trying to decide the difference between the two, we're on a kind of very fuzzy boundary. There are people, for example in cosmetic surgery, there are young women who say that breast augmentation or breast reduction, whichever case they're talking about, is actually not a cosmetic, not an enhancement, but is actually therapeutic because their mental state is altered. Peter Turnpenny, my friend and colleague at Exeter, who's the consultant in clinical genetics, another Christian, talks about a young woman having leg lengthening surgery on the NHS in order to make her taller so she could undertake possibly particular careers. And some people would have regarded that as enhancement, but in this case it was regarded as therapy. So it's a very fuzzy line, and we'll find the same in genetics, I'm sure. is increasing somebody's resistance to the common cold, therapy or enhancement. Now, of course, this raises straight away the idea of designer babies, and the baby then becomes a commodity that fulfills our wishes. I need to say that the idea that you might generate an army is probably not on, because it does require IVF, and as the video quite rightly said, it's an invasive procedure for the woman. She has to be super ovulated by hormones, and then the eggs are extracted. So it's not mass technology. And I think also we must be aware of the science fiction. I talked about a number of features which have some genetic component, but the idea that you might be able to pin those down as single genes is almost certainly wrong. And so, you know, you're a musician, you're a scientist, you're a cricket captain, Crystal Palace football team playing in your waist strip here. Your waist strip, Andy Johnson was still playing for them then. No. You know, I can't get back into the Premiership by genetic modification. So there's a lot of science fiction about it, but nevertheless, as I've said before, there are some physical features of the makeup of all these things which might one day be subject to genetic modification. And the atheist philosopher John Harris uses the following words. You know, if a couple want a curly-haired, brown-eyed, bouncing little girl, why not allow them to choose rather than waiting for God or nature? Interesting, an atheist uses the word God to provide it for them. In other words, let's do it. He says it's no different than paying for ballet classes or trumpet lessons or extra athletic coaching. And I'll take a few more minutes to close. Are there religious views about all this? Well, actually, it's quite hard to characterise, it turns out. The views may well rely on what we think about genetic modification itself. Is the idea of moving genes right or wrong? And more often we find that it's not actually the technique itself, but it's what we do with it. What organism are we modifying? If it's a human, does that make a difference? Or is it a plant or a bacterium? What is the reason for doing it? And so in genetic selection of embryos, the middle part of the talk, are we preventing serious illness, or are we destroying life by not using those embryos for the gene? Is genetic enhancement an acceptable intervention, or are you doing your best for your baby? And just finally, over the last few minutes, since we talked about eugenics yesterday, I think it's worth raising again. Of course, the U in eugenics is the same as the U in euthanasia. It's really all about goods, and euthanasia is a good death. Eugenics is good origins, good breeding, well-bred. No, it doesn't mean that. Well-born. In other words, we're looking for somebody with the right genes to pass on to the next generation. And the idea that we might select people who would be better for society actually goes back to Platonic thinking. So both Plato and Aristotle were keen on the idea that we encourage the better people to breed, and therefore society would be improved. It was a thought experiment, as I think he said this morning, in a sense, because they didn't actually try it out, although noticeably the Spartans used to leave babies on the hillside to see if they'd survive. And then Darwin started to think through this, you know, that maybe the survival of the fittest, that was actually Spencer's term, it wasn't Darwin's term, but we can use the survival of the fittest. Maybe we think about people better fitted for the future of human society. Should we in fact encourage greater reproductive success amongst those folk? And it was then, if you like, formalised by Francis Galton, who was Darwin's cousin and was the first professor of genetics at University College London. And you can go to the Galton Laboratory still in Gower Street, University College. And he basically formalised eugenic thinking. The idea was that we would encourage the fitter to breed more extensively. And the fitter were those who had good moral fibre. They weren't feckless in any way. They weren't the criminal classes. They weren't in any way, as they had said then, mentally subnormal. And of course, it was the people who were encouraged to breed who would make the decisions about this anyway. And when I was a student here in Cambridge in the 60s, you could still find old posters that dated back to the 1920s and 30s, showing farmers distributing by the old broadcasting method that went out in East Anglia years and years before, but only sow the best seed, and they weren't talking about wheat. So there was a kind of eugenic movement in Britain that started with Galton. But actually, it was eugenics here was done in a very amateurish way. In other countries, it was taken on board as on the statute books, and in all these countries we see, USA, Canada, Scandinavia, Germany, Switzerland, on the statute book, that people who were considered not fit to breed were not allowed to breed. And so people were sterilised. The less fit, the morally feeble were sterilised. In fact, don't pass those characters on as if they were built into their genes. The USA has got some tragic examples that, for example, a young woman who might have been a servant in a big house in the Deep South may have been raped by the young man of the house and had a baby. And she would be sterilised because she was morally feeble. And in some cases, if the baby was a girl, the girl was also sterilised. And on a Horizon programme about 12 years ago, they found a woman who had been sterilised as a child because her mother had had her out of wedlock as a result of rape in such circumstances. And in the USA, some 50,000 eugenic sterilisations occurred. The number goes up. When I first talked about this some 5 years ago, we thought the number was 35,000. But talking to American colleagues and looking at the archives, it looks like about 50,000. And it went on to the 50s. In Sweden, the most recent eugenic sterilisation occurred in 1970. Again, of a young woman who had a rather affectless lifestyle, lost her way in life, got on drugs, had babies out of wedlock, and she was sterilised as being morally feeble. So it went on. And so, here's some take-homes. This is a quote and it relates also to transhumanism that Dennis talked about. Science is certainly doing this, including genetic science. It may well be possible to make us clever and strong. What does it mean for our personhood? And Madeline Bunting in The Guardian used this phrase: there's no stop button in the race of human re-engineering. Is it all out of control? And I think there are no easy answers to this, because clearly we can see, if you like, societal goods in trying to prevent suffering. But we've also got the possibility of sliding down this slippery slope that the doctor referred to in the video. Now, here's something that might interest you. We can endeavour it. It's all there. And you also might want to do some reading. Thank you very much.
Thank you very much indeed, John. Well, I'm sure there's still maybe plenty of questions, so we'll start here and then go to the back here.
Thank you. Charles Darwin spent a lot of time talking to pigeon fanciers.
Yes, he did.
Didn't he? And he also, I think, spoke to cattle breeders of Norfolk and other fine counties. What is the difference between breeding a fine sheep or a fine pigeon by those methods and what could be going on here?
Well, if we go to cattle breeding, we can now indeed do that sort of pre-implantation genetic selection if we wanted. But the moment cattle breeding is not that sophisticated, although there's all these things like embryo rescues. So you're suggesting that just sliding it into humans is just a natural progression. Are you? Or?
No, I wouldn't go that far.
Right.
I was asking you that question.
What is going on here is the techniques I've just described in terms of both selecting embryos and modifying embryos, possible right across the mammalian spectrum, and indeed across selecting embryos is a little bit harder in birds, but we can certainly genetically modify them. So in a sense, it's a molecular sophistication of the breeding techniques that cattle breeders, plant breeders, pigeon fanciers have undertaken for hundreds, and indeed in plants, thousands of years. So it brings it down, if you like, to being very specific. We don't have to kind of mix two complete sets of genes together and sort out the ones we want. We can say, I'm going to take this gene stock and put this gene in. Or I'm going to take this gene stock and make sure it's not carrying this gene, so it's precision.
Yes, I think we'll go to the back there and then come forward.
Could I ask you, was your tie specifically designed?
Yeah, an intelligent designer. The tie simply shows a repeating tetranucleotide of the 4 bases of DNA. And it's available commercially, or it was when I bought it, from a company called Science T-shirts of Chepstow.
But it evolved from a cravat.
It probably evolved from a cravat, yes. I haven't seen the genetic changes that...
John gets a commission, I think, for every tie he sells.
I wish that was so.
Other question back, yes.
I've had a chance to hear John Dupre talk about some recent events and all this, so I hope this isn't pulling you off topic at all, but he discusses epigenetics
yes.
and
yes.
DNA cassettes and how at the microbial level there's this promiscuous, gene swapping or DNA swapping going on. I wonder if you have any comments on how that changes our perspective on the whole role of genetics, or how it...
Okay, let's start with the second bit first, gene swapping. It's very clear when we look at our genomes, and Dennis alluded to some of this yesterday, even at the level of a complicated organism such as humans, there has in our evolutionary history been horizontal gene transfer from other organisms. When it comes down to bacteria, they're doing it all the time as part of, if you like, their ecological existence in the environment. They're swapping genes between themselves by sexual and non-sexual methods. So that horizontal gene transfer is built into nature, and the further down the complexity tree you go, the more we can see it happening. In humans, we can only see it happening, if you like, by historical look at our genomes, with the occasional event, such as the incorporation of HIV retrovirus into our genomes, and that's something that we observed in recent history. So yes, that occurs. In terms of epigenetics, it's a very, very good point. It's a point that people who are kind of hardcore geneticists completely fail to appreciate. Epigenetics is the effect on genes of non-genetic characters, such as the environment in the cell, around the cell, and so on. I talked briefly about the epigenetic nature of emotional care. That's an epigenetic effect. It's affecting genes, but it's not a molecular. It's not a genetic event itself. So epigenetics is very important, and it's something people forget. The ultra-reductionists like Dawkins forget it.
Yeah, well, if I may just follow with that, it seems to me, I'm trying to sort out if I'm correct in my perception that this sort of radically changes the whole debate with people like Dawkins who are so committed to the human genetic, passing that they've missed the larger picture of how genes actually work in the huge part of the of the living world, not the human world, that does this kind of gene swapping and DNA cassettes.
He's actually, he actually has a split mind on this, because he talks quite rightly of the extended phenotype. That, when we look at inheritance patterns, for example, in a bird, the extended phenotype is the way they make their nests, the way they forage, the way they lay their eggs. That's all part of what's selected. So he doesn't forget it when it comes to thinking about the world as a whole. He does seem to forget it when it comes to kind of trying to reduce us to a bag of genes. But he's not actually able to do that, as we saw, because he has to have these memes. And although he obviously believes in right and wrong, because religion is evil.
That's right. John, is it worth pointing out also that epigenetic changes to the degree we don't really know how much that can be inherited? I mean, there's some evidence for inheritance with epigenetic change, but exactly how many generations that might pass through is yet, I think, an open question.
Well, indeed. I mean, maternal imprinting, that is, if you like, things that have happened to the mother epigenetically, we now know can be passed through at least one and possibly 2 generations, even in complex mammals. So I think it was the women who were starved in the Netherlands during the war, and their babies were low birth weights, inevitably, but their grandchildren, the children of their daughters, were also low birth. That's clearly an epigenetic effect.
This sounds like the inheritance of acquired characteristics.
Well, it's not. No, it's not the Markinism, because it will eventually disappear. What it is, it's effectively a non-genetic character on the expression of the inherited component, the gene.
Although I must say, Dupre has talked about the revival of...
I know he has. I mean, John is a colleague of mine, so I see him quite often.
There was a question here first, actually, and then we'll go with here. Yes.
I speak as a retired GP. Getting back to the philosophical question, do we or don't we use this? It's valuable to look back in history and to see how we doctors, society, objected strongly to anaesthetics.
Yes, indeed.
Because it took away the pain of childbirth, which in pain surely bring forth children. We now accept that entirely. We Protestants anyway have accepted family planning, manipulation of that. I've done it. Is this any different from what we've gone through eons, and doctors have played God all the time and are forced into playing God?
You mean, for example, selection against embryos carrying particular genetic conditions?
And on, because it's a process.
I think for me, there are some genetic conditions which cause the child to be born such a difficult and unpleasant and short life that I would see selection against those conditions as simply being yet part of our medical care for human beings. There are others where I think actually we are in the area of not quite, we want perfection for the sake of it. And this is what I mean about the narrowing down of our ideas of normality. So I think there is a gradation, and where you place your stop boundary, I think is very difficult. This isn't a case of going up to a fence and thinking about jumping it and then going to another fence. I think we are here on a slope, and where medical care turns into sheer hubris, I do not know, and it might be for each individual parent and doctor to decide that.
Yes, I think actually a question there and then there. Yes, so.
I just wondered if there was any complications in this that might relate to mitochondrial DNA.
Yeah.
The,
Mitochondria, for those who are not biologists, are the organelles in our cells, the subcellular compartment, one of whose jobs is to provide the energy for driving that cell's life. Sometimes it's called the cell's battery. It does other things as well, which people forget. But it has about 20 genes. It relies entirely on the rest of the genome for its life, because it can't exist with 20 genes. But it does carry some inherited characters. For example, we had a student some 5 years ago who went blind during his courses as an undergraduate because one of his mitochondrial genes had a mutation which caused a late onset failure to provide enough energy to fire the optic nerve. So mitochondria do contribute to our genetic makeup. I would say that in terms of genetic selection, I don't think their presence makes any difference to the principles. We can detect that mitochondrial mutation. So it may do in what I'm going to talk about this afternoon, which is stem cells, but I think in terms of straight genetics, it doesn't make any difference at all. Just we acknowledge that they're there. They make a tiny proportion of our overall set of genes.
And then the question, yes. Yes, please, yeah.
Right in the middle.
Yes, indeed, your hand. Yes, sorry, yes.
Yeah, you talked about how it's not genes are us. There's this recognition that there's a limit to how much we're genetically determined.
Yes.
Beyond that, there's whatever in the environment. I just wonder if that recognition actually makes it more likely that pre-implantation screening will become widespread, because that means the slippery slope has an end point, doesn't it? And actually people will say, well, look, there's only a certain number of things that we can control, and it's not quite as much of a Pandora's box as you thought it was, and that makes it more likely it'll become acceptable.
I think that's certainly one viewpoint, that the slippery slope will have a stop point. But what about these areas such as intelligence, where there is an inherited component? There is an inherited component, but there are so many other components. And so what you do is you enhance the inherited component to make sure the environmental and social and learning factors have got the best material to work with.
Yeah.
So I think as the doctor in the video plays a very large part, are going to be the hardest to deal with ethically. So maybe the slippery slope, it'll sort of, but I think it's.
Yeah, you said the genetic difference between any two males is between 0.1% and 1%.
And between any two females.
Total genome or the functional genome?
No, that's the total genome.
And what is the significance of the fact that the vast majority of
I hate to use the word jump. What our colleague is talking about here is that if we look at the coding sequences in our genome, the sequences that actually provide those recipes I was talking about, they only make up a small proportion, and much of the rest of our DNA is made up of either controlling sequences, such as the front and the back end of genes, and much more. It's also made up of repetitive sequences, such as the sequence of my tie, the tetranucleotide. And some of the differences we're talking about are differences in the non-coding DNA. So it's basically between 1 in 100 and 1 in 1,000 bases are different, and some of those differences are in the coding DNA and some are in non-coding DNA. It's the differences in the coding DNA that provide actual phenotypic differences, but they're quite small. So they're spread right through the genome. A classic example would be in some of the repetitive DNAs, you can generate a unique genetic fingerprint for you by the way that the repetitive DNA can be cut by enzymes, and this is used forensically.
What could just add a little point there? I mean, one of the interesting things in Craig Ventner's genome was they actually sequenced both chromosomes, which hadn't been done before. So now we have a human individual where both chromosomes have been sequenced. And actually the differences between the two chromosomes are more than people have realized. At the moment, I think it's fair to say that John hinted at this, the pendulum is swinging. We're a little more different genetically than we thought.
Absolutely. I mean,
Francis
Francis is 0.1%, but it's certainly, we think the average is now near 1%.
Yes.
And certainly Craig's maternal and paternal set were more different than he was expecting.
Yes. Okay, we'll go to someone who has to ask you. Yes, please. Yes. Sorry, Dan. Yes. No, that is you first. No, we'll have to hear in the back of your head.
This isn't going to be a crass question, but I wondered if there's a kind of a meta question that's going over all the top of this. Is obvious, and that is, what is a human? And if what I've found in my conversations with the people that I work with in the faith community that I'm part of are, we shouldn't do that because that's a human being. And that's the big question that's over all of this. And what I found in discussions with people, it's when those questions, society seems to move the boundary of that for the convenience often of the process or the technique that's being advocated. So the question about whether an 8-cell embryo is a human being or not. Whereas I find myself drawn towards the notion of accepting that the embryo is a human life, and then having to face those hard choices, having accepted that it is, because that seems to be a more honest. Now I could say, no, it isn't a human life, and choose another point when it becomes a human life, but even there, at that point, we still have to make those questions.
I'm very postmodern here. Yeah. It's more honest for you.
Thank you.
I, as a biologist, find it really hard to ascribe human personhood to the early embryo. Human life is a word I will use, but human personhood I find it very difficult to suggest the 8-cell embryo is a human person. And I don't think actually in the scientific community or in the medical community, the views of when human life begins, which is the way the question is always rather badly asked, has changed as a result of our understanding of genetics. I think our sense of wonder at the early stages has increased as we see that we go from no commitment at all to after implantation we see cells begin to be committed. But I don't think that we have changed overall, except maybe by secularization, our view of the early human embryo. I mean, IVF was invented in 1968, and it inevitably leads to some embryos not being implanted. So you could ask about what is the status of those that aren't implanted, but you could equally ask what is the status of those that in nature fail to establish a pregnancy after fertilization, which in humans is a remarkably high percentage. It's something like 70 to 80%. And so, you're dealing with the same issue. One is a natural wastage, and one is a medically selected wastage. But I don't see any difference in the moral status of those two sets of rejected embryos.
And who rejected them? Sorry.
Well, that's right. That may be the crux of the matter. You know what? This is why some people talk about playing God. You know, God decided which embryos would implant in nature. We're deciding which embryos would implant in IVF, and that's certainly. I mean, I don't look at it that way, but you know, clearly you've represented a viewpoint which is out there.
I think it's Hillary was next, and then we'll go to the back. Yeah.
Okay, well this is a question that I've been mulling over, and then when you talked about having sequenced both sets of chromosomes, it maybe highlights it, and it arises from a conversation, almost debate, even argument I was having with my mother last night as to the nature versus nurture kind of lines, but actually it was conceptualizing this thing that we often reflect on if we have more than one child, about why your children are so different and yet you can see bits of you in them, and particularly relating to bits of to whether your children are sociable or not.
Yes,
and some of mine are more introverted than others, and so on. And so, I mean, does the fact that the difference between the two sets of chromosomes is perhaps greater than was assumed help explain some of these more personality, sort of less quantifiable differences between family members?
I don't think we need to call on the Venter data to actually explain that at all, because when you think about how gametes, the germ cells, are formed, with the recombination of DNA that occurs in a process called meiosis, which is the DNA replication system that generates, and you see the mixing between the different pairs of chromosomes that occurs during gamete formation, I think the differences are much more explicable because of that than they are. We don't need to invoke the difference between the maternal and the paternal chromosomes to do that. And of course, personality is a very interesting thing. That, you know, it does appear that elements of our personality do come through our genes, and maybe that shouldn't surprise us 100%, because you know the brain depends on electrical and biochemical signaling, and the biochemistry is obviously regulated by genes, and the structures that provide the electrical signaling are regulated by genes. And so I think it's inevitable that some elements of the way we are personality-wise will be at least partly in our genes. But we also know that personality can be modified extensively by the social background and by life experiences and so on. I talked about the children in Romania who've got almost no sociability at all, and in fact great areas of their brains fail to develop.
There was a question right at the back,
I think you're actually addressing it, and I'm not putting it together or smart enough to see how it flows together. So let me reformulate the question slightly. We've talked about a 0.1 or 1% difference. We've all heard the statistic oft repeated about how marginally different we are from apes genetically. You've talked about how we've gone from, you know, an expectation, well, I actually didn't say the high number, but as I recall, they were expecting several 100,000 genes.
We were expecting between 80 and 100,000.
80 and 100,000. Yeah. We actually found some 26,000.
Yes.
It seems to suggest that we're looking in the wrong place, that this isn't as dominant an issue in who we are and how we came to be and what we might be in the future as we once thought. I wonder what your thoughts are on that.
Yeah, I mean, let's go back to your starting comment about the similarity to the great apes. And I would recommend people to go and read Dennis Noble's book, The Music of Life. You know, once you've got past his annoying habit of making sure that you know how cultured a person he is. It's actually a very nice book, because it points out the falsehood of implying that adding 7 genes simply adds 7 functions, because we know, albeit very, very shallowly, that genes interact with each other, sometimes in cassettes and sometimes not in cassettes, during development and during life. And if you add 7 genes to 26,000, the number of possible interactions you've added is actually a very large number. So the number of genes per se isn't all there is. And the fact we've got 26,000 means that we've got a huge number, I mean it's a vast number of possible genetic interactions. And some of those interactions will actually be apparent as so-called genetic characters. But nevertheless, we've still got the problem. There are some actual pinpointable genetic conditions which must be and must all come from one gene. And we now know mechanisms for generating more than one trait from one gene. And in fact, there are at least 4 different mechanisms for doing that. And I think the record so far is that we know of a gene that is active in early chicken development that codes for 11 different proteins by different routes. So that's getting on for being, quite a versatile gene, that one. The gene I work with, it was a moonlighting gene, I think, it's only got 3 different functions, of which we know 2 for sure.
Yes.
John, what do you think are the key questions that your work poses for theology?
Well, my work has been on plant and yeast genes involved in regulating DNA replication, and so the question I would pose to theology from what I've found out is, wow, why did the Creator do it this way? It looks much too complicated. When I started my work on DNA replication as a postgrad and then a postdoc, we had a very simple idea, a bit more complex than Watson and Crick's idea of how it worked, but nevertheless quite a simple idea. And it turns out to be such a vastly complicated process, you wonder how on earth our cells managed to do it. So my theological question to the Creator is in a why. But in terms of the more general viewpoint, I think what the theological question it raises for me is how far should humankind change the genetic makeup of a range of living organisms? And as far as I'm concerned, I'm entirely happy for us to do that with crops. So, I don't apologise for my espousal of plant genetic modification. But when it comes to mammals, we begin to have doubts. For example, we'll have doubts in non-human mammals about are we increasing the commodification of farm animals by changing the genetic makeup? Are we affecting their telos? When it comes to humans, are we beginning to affect who or what they are as individual human beings? So those are the sort of questions that I think we're talking about.
Good. Well, I think, if it's a fairly brief question, perhaps we'll take one more then.
I think very, very brief, actually. Just following on that point. Hey, question's gone, sorry.
Gene got switched off there. Yes.
Pre-lunch epigenetic modification.
Absolutely, it was. I think lack of glucose setting.
We all have that. Actually, first of all, we'd like to thank John very much, I think, for a great session.